Hello everyone, today's lab will be about Gram stain. First, what Gram stain is one of the most fundamental and widely used staining technique in medical microbiology. This technique differentiates bacteria into gram-positive and gram-negative group based on difference in the structure and composition of their cell walls, particularly the thickness of the peptidoglycan. A stain or dye is a chemical substance that has the ability to bind to cellular structure and impart color to them. Since most microorganisms are transparent under the light microscope, staining is essential to make them visible and distinguishable from the background. Why a stain have color? A stain contains a special chemical group called chromophore. Chromophore are part of the dye molecule that conjugate double bonds. These structures absorb visible light, which give the dye its specific color. Without chromophore, a dye would be colorless and useless for microscopy. How a stain bind to microbial cells? Stain molecules attach to bacterial cells through different types of chemical interactions. Ionic bonding, which occurs between oppositely charged ion. For example, positively charged dye binds to negatively charged bacterial cell wall. This is the most important mechanism in gram staining. The second one is covalent bonding. A strong chemical bond between the dye and cell component, and using special stain, example, flagella stain. And then we have hydrophobic bonding, which occurs between nonpolar region of dyes and cell structure and help stabilize dye attachment. And now we have the classification of stains. Stains are classified based on the electrical charge of their chromophore and how they interact with cellular component. The first one is acidic dye, negatively charged. Acidic dye carry a negative charge. They are attached to positively charged component of the cell. The background is usually stained while the cell remain unstained, also called negative staining. Example, eosin and picric acid. Common use: demonstrating cell outlines, background staining like capsule demonstration. The second one is basic dye, positively charged. Basic dye carry a positive charge. They are attached to negatively charged bacterial cells due to nucleic acid and acidic cell wall component. This dye directly stain the bacterial cell. Example, crystal violet, methylene blue, safranin, malachite green. Clinical importance: most bacteriological stains, including the gram stain, use basic dye. The third one is neutral dye, formed by combining an acidic dye with a basic dye. Contain both acidic and basic radicals. Can stain different parts of the cell simultaneously. Staining techniques are classified according to their purpose and the number of dyes used to visualize microorganisms and their structure under the light microscope. We have simple staining, which involve the use of single dye to stain bacterial cell, to observe basic cell morphology and also to determine shape, size, and arrangement of bacteria. Commonly used dye is methylene blue, safranin, carbolfuchsin, and crystal violet. And then we have differential staining. Differential staining, which uses two or more dyes to distinguish between different groups of bacteria based on their structure and chemical properties. The purpose of this technique is to differentiate bacteria into distinct categories. Example, gram stain differentiate gram-positive and gram-negative bacteria. Acid-fast stain differentiates acid-fast and non-acid-fast organisms. The third one is special staining. Special staining technique are used to demonstrate specific bacterial structure that are not visible with simple or differential stains. The purpose of this technique is to study of special structures under the light microscope. Example, negative staining, capsule demonstration, Schaefer staining, endospore staining, flagellar stain, demonstrate flagella. Appears dark line with silver or red with carbon 14. The Gram stain technique developed by Hans Christian Gram in 1884 and later modified by Hacker in 1921. It is one of the most important and routinely used methods in medical microbiology. The principle of Gram staining. The Gram staining differentiates bacteria based on differences in their structure of their cell walls, particularly the thickness of the peptidoglycan layer, as we said before. Classification of bacteria by Gram stain. Using this technique, bacteria are divided into two major groups: gram-positive bacteria and gram-negative bacteria. Gram-positive bacteria retain the primary stain, which is crystal violet, appear purple or violet under the microscope, have a thick peptidoglycan layer which traps the diiodine complex. Gram-negative bacteria do not retain crystal violet. After decolorization, take up the counterstain, usually safranin, appear pink or red under the microscope, have a thin peptidoglycan layer and an outer membrane. Gram-positive bacteria are characterized by a thick and complex cell wall structure, which is responsible for their purple appearance after gram staining. The thickness of the peptidoglycan layer. The cell wall contains a very thick peptidoglycan layer. Approximately 90% of the cell wall is made up of peptidoglycan, which provide rigidity, shape, and protection to this bacterial cell. Extensive cross-linking. Peptidoglycan strands are extensively cross-linked. This dense network traps the crystal violet iodine complex, which prevent dye loss during the decolorization step. And the third one is lipoteichoic acid or teichoic acid, which are embedded within the peptidoglycan layer. Lipoteichoic acids anchor the cell wall to the cytoplasmic membrane. Cytoplasmic membrane, which lies beneath the peptidoglycan layer, contains structural and enzymatic proteins involving transport, metabolism, and energy production. Unlike gram-positive bacteria, gram-negative bacteria have a complex multi-layer cell envelope that is structurally and chemically different from gram-positive bacteria. This structure explains their pink appearance after gram staining. The outer membrane, which present only in gram-negative bacteria, lies external to the peptidoglycan layer, act as a protective barrier against antibiotics, detergent, and dye. Lipopolysaccharide, major component of the outer membrane, composed of lipid, A, endotoxin, core polysaccharide, and O antigen. The third one is thin peptidoglycan layer. Accounts for only 5 to 10% of the cell wall. Located in the periplasmic space, has minimal cross-lining, and cannot retain the crystal violet iodine complex. The periplasmic space, which between the outer membrane and cytoplasmic membrane, contains enzymes and transport and carrier proteins, important for nutrient processing and resistant mechanism. Purine proteins, which channel forming proteins in the outer membrane, allow small hydrophilic molecules to pass into the cell, and regulate antibiotic entry. The last one is cytoplasmic membrane. Typically, phospholipid bilayer contain carrier and nutrient-binding protein, site of metabolic and transport activity. As you can see in this figure as well, the gram-positive bacteria is on the left side and gram-negative bacteria on the right side. Gram-positive bacteria have a thick layer of peptidoglycan, the green layer, around the cell membrane. This thick layer helps keep the bacteria strong and give them the ability to hold onto certain dyes in a lab test called the Gram stain. Gram-negative bacteria, which have a thin peptidoglycan layer, locate between two membranes. They have an extra outer membrane, yellow and purple layer, outside the peptidoglycan. The space between the membrane is called the periplasmic space. They also have porins in the outer membrane that allow some molecule to pass. The materials that we need for a gram stain is we need a clean grease-free slide, microscope with oil immersion objective, inoculation loop, bacterial culture, Bunsen burner, and gram stain reagent. We need crystal violet, gram iodine, gram decolorized, which is 95% ethanol or acetone, and safranin. Before staining the bacteria, we need to prepare the smear. How the smear is going to be prepared? First, we are going to bring a clean cloth slide, then add 1 to 2 drops of distilled water with help of inoculated loop, and then heat fixation for 10 to 50 seconds, and take a colony of bacteria from the culture and with the help of inoculated loop and mix it with water and let it dry. After that, we are going to do the gram staining procedure. The gram staining protocol. First, we keep a slide in staining rack. We added crystal violet and wait for 1 minute. After that, we wash the slide with distilled water or low running tap water. After that, we added gram iodine and wait for 1 minute. Then we wash the slide with running tap water or distilled water and added gram decolorized, which is 95% alcohol. Wait for 20 to 25 seconds. Wash a slide with slow running tap water and then added safranin counter. Wait for 30 seconds, which wash with slow running tap water, allow to air dry and observe it under oil immersion. As we said before, the gram-negative bacteria appears pink under the microscope and gram-positive bacteria appears purple under the microscope. And then we have the theory of gram staining, which attempt to explain why gram-positive bacteria retain crystal violet and gram-negative bacteria don't. Two classical explanations are highlighted. The pH theory. The cytoplasm of gram-positive bacteria is more acidic compared to gram-negative bacteria. Crystal violet is a basic, positively charged dye. Because opposite charge attract, the acidic cytoplasm of gram-positive bacteria retain the basic dye for a long time. Role of iodine. Gram's iodine acts as a mordant, fixative. It combines with crystal violet to form a crystal violet iodine complex. This complex is large and insoluble, so it becomes trapped inside gram-positive cells, especially during the decolorization steps. And then we have the magnesium ribonucleic acid. Magnesium ribonucleate or magnesium RNA and basic proteins are present in higher concentration in gram-positive bacteria, especially near the cell membrane. These compounds have a strong affinity for basic dyes, like crystal violet. During staining, crystal violet binds tightly to magnesium ribonucleate. Then, during decolorization, alcohol removes lipids and magnesium RNA from gram-negative bacteria. In gram-positive bacteria, magnesium ribonucleate remains intact and continues to retain the crystal violet iodine complex. Cell wall theory of gram staining. The difference in cell wall structure between gram-positive and gram-negative bacteria determines dye retention during gram staining. Gram-positive cell is very thick peptidoglycan layer, tightly cross-linked structure, and minimal lipid content. Mechanism of dye retention: crystal violet and iodine form a large crystal violet iodine complex inside the cell. During decolorization, alcohol or acetone dehydrates the thick peptidoglycan, causing shrinkage of pores. The shrunken pores trap the crystal violet iodine complex, preventing its escape. Gram-negative cell wall, which is a thin peptidoglycan layer, high lipid content, presence of lipopolysaccharide, outer membrane, loosely cross-linked structure. Alcohol or acetone dissolve the lipid-rich outer membrane. This creates large pores in the cell wall. The crystal violet iodine complex escapes easily from the cytoplasm. Cells become colorless and later take the constrained safranin. And then we have gram-positive and gram-negative cocci and bacilli. The gram-negative positive cocci, which is Staphylococcus, Streptococcus, and Enterococcus. Gram-positive bacilli, Bacillus, Corynebacterium, and Clostridium. We have then gram-negative cocci, Neisseria and Veillonella. Gram-negative bacilli, we have E. coli, Klebsiella, Proteus, Enterobacter, and Pseudomonas. Use of gram staining. It is used to differentiate bacteria into gram-positive and gram-negative bacteria, as we said before, and identification of bacteria, and then to start empirical treatments.